Hi, I'm Jamie Batcher, instructor for A&P 2. We are continuing on with our lecture on kidney physiology. We just talked about regulating GFR, or glomerular filtration, right? Say that 10 times fast. We're going to go on and start talking about reabsorption and secretion and how all of that works in our nephrons. That's the questions that we just left off on. Let's talk about the reabsorption process. So remember, there's three things that are happening in the kidneys. Filtration, which we just talked about, reabsorption, we're going to talk about now, and secretion. So with reabsorption, as I mentioned before, the majority of this is happening in the PCT, right? Remember, that glomerulus is filtering out a lot of stuff. 99% of it's coming right back in. Glucose, amino acids, organic nutrients, all taken back into the peritubular fluid, then into the peritubular capillaries, right? Sodium, potassium, bicarbonate, all of these things right back in. Water, right back in. So we kicked it out and now we're taking it back. All goes into the peritubular fluid and then into the peritubular capillaries, which connects up with our cortical radiate veins, right? So there you see this whole process and how it's happening. Look at this tubular fluid, right? This is that pre-urine here. This is a cross-section of our PCT. Notice that it's increased surface area here. Notice these villi. Why is that? Well, anywhere where we're reabsorbing, we want to have increased surface area to allow for that increase in reabsorption rates. And that is how your PCT is put together, right? All of these villi are there to increase the surface area so all of that stuff can come back. So from our tubular fluid, glucose taken right back in. That's why we don't eliminate it in the urine, right? Osmotic water, right on through. Ions, sodium, potassium, bicarbonate, all of those things. Typically, they're mostly counter-transported, so it's like an exchange, like sodium for potassium, right? Or potassium for bicarbonate, or sodium for hydrogen. So there's these little counter pumps. We'll talk about those in a little bit. But either way, they get reabsorbed from the tubular fluid. They go through the peritubular cells, into the peritubular fluid, and then into the peritubular capillaries, right back into the bloodstream. Secretion is the opposite. This is movement of water and solutes out of that peritubular fluid into the tubular fluid. Very small percentage here is actually going to be affected by this. Only 15 to 20% of the filtrate volume that gets to the DCT is actually going to be excreted. So it's a combination of reabsorption and secretion and the DCT that's ultimately that's going to contribute to our solute composition in this tubular fluid or filtrate. So sodium ions are typically reabsorbed through exchange pumps. Hydrogen ions are exchanged for sodium ions, again, exchange pumps there. And carrier proteins are there to secrete any drugs or toxins, put them into the urine so that our body can get rid of them. So notice here the difference of our tubular cells, we don't have those villi here. And again, the exchange pumps, sodium and potassium move opposite, sodium and hydrogen move opposite. So depending on what your body's trying to do, it might be pumping out or pumping in, one or the other. One thing I will say that's important, it's going to be important later on in our next chapter, is notice the relationship between potassium levels and hydrogen ions. Potassium and hydrogen ions like to go together because they both move opposite of sodium. So if you have a lot of pH in your body, you might also have a lot of potassium in your body. I say a lot of pH, that's not what I mean. A lot of acid in your body, a lot of hydrogen ions, because remember, acidity is actually a low pH, right? Sorry, I may have confused you there. All right, you can pause the video here, go back and listen to what I should have said instead of what I did say, because I didn't mean to confuse you there. Make sure you understand how reabsorption and secretion are different and their relationships to the reabsorption and secretion of hydrogen ions, sodium, and potassium, how they are all interrelated as well. Let's move on and talk about what goes on in the nephron loop. So we talked about the processes in general: filtration, reabsorption, secretion. There's something important that happens in the nephron loop that it kind of blows my mind. It's really, really cool. So remember, in the glomerulus, you have mostly filtration, PCT, mostly reabsorption. DCT, you have variable secretion and reabsorption going on based on other factors, right? In our nephron loop, which again is between the PCT and the DCT, you have this mechanism working called countercurrent multiplication. Countercurrent multiplication. It's kind of like countercurrent exchange, right? Where you have these two things that are moving in opposite directions that are going to affect each other. So the thin descending and thick ascending limb of our nephron loop are very close to each other, and there's peritubular fluid in between. There's an exchange that takes place between these limbs, and it's called countercurrent multiplication. So remember that thin limb is mostly responsible for reabsorbing water. That thick limb is reabsorbing solutes. So let's think about how that works. As our fluid is moving through our thin limb and all of this water is being sucked out of it, this peritubular fluid is being super concentrated, right? Because all of the water is being reabsorbed. By the time this tubular fluid gets to the ascending limb, we have a super concentrated tubular fluid amidst a peritubular fluid that has been diluted. Why has it been diluted? Because on the descending side, all of the water was being reabsorbed. So you have water being reabsorbed from one side and solutes being reabsorbed from the other. So the solutes are going to be reabsorbed by our thick descending limb into the peritubular space because the surrounding fluid is diluted. What does that do? Well, it's going to dilute and increase the solute concentration of the peritubular fluid, which is going to then do what? Pull more water out of our descending limb, which is going to what? Pull more solutes out of our ascending limb, and so on and so on and so forth. So it multiplies itself. As one is reabsorbing more water, that's going to make the other reabsorb more solutes and more water and more solutes and more water and more solutes, which is why juxtamedullary nephrons are very, very long. The nephron loop is very, very long, and that vasa recta is right there to help reabsorb and take all of these things back into the blood. Remember, the vasa recta is important in juxtamedullary nephrons because it's super concentrating the urine, right? So the thick ascending limb is actively transporting sodium and chloride, those two major ions there, out of the tubular fluid. It's impermeable to water, so water cannot be reabsorbed or secreted on this thick side. The tubular fluid solute concentration is going to decrease, and the peritubular solute concentration increases, which again is going to pull more water out of that thin side. Here's all of it in action. You can see this is the thick ascending limb, and sodium and chloride are our major ions that are being reabsorbed here, right back into the peritubular fluid, which is going to concentrate that peritubular fluid, which is then going to pull more water out of the descending side, which is going to pull more solutes out of the ascending side, and so on and so forth. So that nephron loop uses that countercurrent multiplication to concentrate our urine. The osmotic concentration of the peritubular fluid increases as it moves through because it's being concentrated. The thin descending limb is permeable to water and impermeable to solutes. So the tubular fluid solute concentration increases as it moves through. So all the water is being taken out and solutes are left, the solute concentration increases. Here you can see it in actual numbers. If you're a numbers person, you want to understand how this works. Going into our nephron loop, you have roughly a solute concentration of around 600. As you're pulling more water out, it's getting more and more and more concentrated until around the end, you're at 1200 or so, right? Then on our thick ascending side, solutes are going to be reabsorbed, and we get back up to 300 to 100, right? Sodium and chloride are going out into that peritubular space because it's been nice and diluted from our descending limb. So how is our urine concentration made up? Well, the water is reabsorbed from the tubular fluid and then in the DCT and collecting duct. It all goes right back into our tubular fluid, into the peritubular capillaries. It's going to increase the concentration of solutes in that tubular fluid, particularly urea, very high concentration of urea in your urine, hence urine, right? That's kind of how it gets its name. The tubular fluid that reaches that papillary duct has a urine concentration of around 450 milliosmoles per liter. Super concentrated. right? To take a look at it here, our thin descending limb, leaving water, our thick ascending limb, all of our sodium and chloride being reabsorbed. And since sodium and chloride has been reabsorbed here, our solute concentration is back down to around 100, but in our collecting system in DCT, all of the variable hormones like aldosterone and ADH can play a role, helping to reabsorb more water back into the body, super concentrating the urine. You can pause the video here, go back and review this section, talking about how our countercurrent multiplication works. It's super cool. I think it's cool. I'm a geek. All right, let's talk water regulation. So the amount of water that's being reabsorbed is going to affect the amount of volume, the volume of urine that we produce every day, as well as the osmotic concentration of our urine. So there is something called obligatory water reabsorption. This is where PCT and the descending limb of the nephron loop, there's no way to control that. It's just naturally going to happen, can't be prevented, right? The rate can't be adjusted. This is the majority of our filtrate that's reabsorbed is going to be reabsorbed in an obligatory fashion. There's also facultative water reabsorption. This is under the influence of hormones. So this is the DCT and the collecting tubule, adjusting that volume of urine and concentration of urine to get that kind of last little change before it's eliminated. So obligatory is pretty much most of our nephron except the DCT and collecting duct. How is our urine volume regulated without ADH? Well, no water is reabsorbed in the DCT or the collecting tubule without ADH. There's no facultative water reabsorption without ADH. The fluid is just going to become part of your urine. So you're going to produce a very large amount of very dilute urine. This is when you have pee that's like clear, right? And you know this, right? You drink a lot of water, what's going to happen? You're going to pee it all right out, right? So there you go. No antidiuretic hormone, no ADH, and so we have water reabsorption in the PCT and in the descending limb, but not in the DCT or collecting duct. So we have this very dilute urine that's being eliminated. When ADH is released, and we have the presence of ADH, little aquaporins are going to appear in our DCT and collecting system. What is an aquaporin? It's a protein. So the hormone, follow this, the hormone that's released, ADH and or aldosterone, is going to go into our tubular cells. It's of our DCT and collecting system. Our tubular cells are going to undergo protein synthesis, right? DNA, transcription, translation, remember all that from genetics or biology? They're going to simply make aquaporins under the direction of that hormone. These aquaporins are going to insert themselves in the plasma membrane of the DCT and collecting duct. And now our DCT and collecting duct cells can reabsorb water and or sodium as needed. So the water permeability of the last tubular segments is going to increase under the direction of ADH. It's very small concentrated urine is going to be produced when ADH is present. There you see it, ADH is going to reabsorb all that water, and so we get a very small volume of super concentrated urine. Normal urine volume is about 1,200 milliliters a day, a little over a liter a day. Variable osmotic concentration around 1,000 milliosmoles per liter. Obviously from person to person, day to day, it's going to vary based on your diet, your exercise level, your activity level, etc. Kidneys alter your the output based on what your body needs, right? Which is why I said at the very beginning of this lecture, of this chapter, that your kidneys are one of your main homeostatic mechanisms in your body, hands down. So some characteristics of urine, right? An average pH, it can be acidic, it can be alkaline. Typically it's a little acidic, average is around 6.0. Specific gravity, this is kind of like density almost, is how heavy it is. It's much like water, a little bit heavier than water. Has an osmotic concentration, again, around 1,000, variable though. Mostly water, up to 97% water concentration. Volume is going to vary based on your intake. Typically a clear yellow color is what it should be. Should not have a specific odor, but again, with composition, depending on what you may have ate or may have drank, your urine may have an odor to it, and it should not have any bacteria in it. It's sterile. Normal healthy urine is sterile. It can grow, it can serve as a growth medium for bacteria, but that's only after time. You can pause the video here, go back and review this section on urine volume and concentration. We're going to keep moving and talk about renal function as a whole. So, overall review, our filtrate has the same osmotic composition as our plasma, right? When in that renal corpuscle, when filtration first occurs, whatever's in the filtrate is the same stuff that's in the blood, minus a few plasma proteins, right? Same composition. In the PCT, ions, organic nutrients, all of those things are removed from the tubular fluid. Water's going to go just through simple osmosis. This is going to reduce the tubular fluid volume, but keep the tubular fluid and paratubular fluids isotonic. So there's not going to be a concentration difference inside the tube or outside the tube. It's the same until we get to the nephron loop, right? So the PCT and the descending limb of the nephron loop, this is where obligatory water reabsorption is going to concentrate that tubular fluid. So this is looking at overall renal function. Starts there around 300, is going to be super concentrated as we go down our nephron loop, descending limb, and this is because of all that increased water reabsorption from our nephron loop. So the ascending limb of our nephron loop, this is where active transport is going to take over and pump sodium and chloride out of the tubule. Water can't follow on this side. It's going to lower our osmotic concentration of the tubular fluid. The DCT and collecting systems can make adjustments based on hormone input. The vasa recta of our juxtamedullary nephrons is going to maintain the concentration gradient of the medulla of the kidneys by absorbing solutes and water from the tubules directly into the systemic circuit. So here you see that ascending side, the DCT, and the collecting duct, all variable reabsorption of water and solutes, but that ascending limb is reabsorbing sodium and chloride. Here's the whole thing put together. I suggest spending some time with this, looking at the key at the bottom, figuring out where things are going, why they're doing it, what happens when that doesn't work. Think about some scenarios, look at this and say, okay, what if aldosterone wasn't present, right? What would happen if you didn't have a DCT, right? What would happen if you didn't have a descending limb of your nephron loop, right? Things like that. See if you can play out a couple different scenarios, because when you think about those what if questions, you're really checking your overall understanding. You can, I think I'm going to stop the video here so you can go back and review this section. We might have one or two more little videos of this chapter. So we're going to go into what happens in renal